Welcome one and all. Eric Smith, Origin of Life researcher, recently gave a presentation, and we're going to be going over that today. It's entitled Information Gateways to Life. I've got Nestlig20 with me here. He was excited to go over this video, so we're going to check it out. And Nestlig has some commentary on it for us, and any kind of haphazard thoughts that I have along the way, I'll see if I can share. So with that, how's it going, Nestlig? What you got going on?
I'm very excited to go over this talk. Like I've been ever since I have seen some of the earlier videos or presentations by Eric Smith. He has a very elegant way of talking and presenting that makes sense. And it's also one of the things that I have been I have changed my view around the origin of life. Like I was really into the RNA camp, you know, like, oh, life started by RNA and RNA built itself up from there. But now I'm more into more into the geochemistry, the self-organizing nature of central metabolism. But now Eric Smith is also going to talk about the more of the higher order structures like RNA, proteins, and the translational system. That is also a very important feature of the origins of life, of course. So, do we go straight into the video, or do you want to make your last note that you want to talk about before we went live? What do you think?
No, other than to just say that I share your opinion about Eric Smith. I think he's an excellent presenter. He's a really smart guy. I've gained a lot from listening to his presentations. I still have to make my way through the majority of his book, but everything that I've come across from him is really well thought out and put together. So yeah.
And just in case this video goes on YouTube, the book that Drayce is referring to is called "The Origin and Nature of Life on Earth: The Emergence of the Fourth Geosphere" by Eric Smith and his colleague Harold J. Morowitz. It's a very good book. Though it's a very long book and a very detailed book. It's a very difficult book to go through, but it's but because it's so detailed, it's a very good read nonetheless. But yeah, without further ado, I think I will start the video. What do you think?
Take it away.
All right.
The final one of this session before lunch is Eric Smith. And he's from the Georgia Institute of Technology and the Earth Life
Maybe I can skip towards the start of the talk. Let's see here. And there we go.
Okay. Talk today is supposed to be about the origin of life, and my point of view will be that life is not one thing. It's an accretion of many things that are different in kind, and the origin of life was not one event. It's a collection of stages, and some of these stages, I want to argue to you, were law-like and predictable from first principles, and those are the things that I'm calling gateways. But to start all of this, let me go back a step and tell you the practical.
Okay, I think I have some notes already. Like he's talking about how some aspects of, like, there are many aspects of life, of course. Like even in the case, like even though I call myself more on the side of the metabolism first, so-called metabolism first camp, I still have to acknowledge that, of course, life isn't just metabolism. It's many other systems are more important. So we need to keep that in mind as well. And I would say you also agree with that, Drayce, right?
Yeah, and I also think it gets into something that a lot of other, metabolism firsters or alkaline vent proponents talk about: life as an emergent structure, and it's kind of a way to differentiate it from older ideas of spontaneous generation, where you have life pop into existence all at one time, fully made, rather than this step by step derivatization process, where you get like increasing complexity that approaches a threshold, and once you pass that threshold, you can start talking or at least using terms like life.
Right. It's a very, like, I think what's his name again? Shostak. I think like he has a paper. I don't remember which title it was. And I think in one of the talks by Eric Smith, not this one but another talk, he mentions that paper, and Shostak emphasizes how drawing a line where, oh, this is life, this is not life, or this is life, this is not life, like drawing a line like that, doesn't really help in the origins of life research, because there are many different aspects, as Eric Smith already noted. There are many different aspects, and all of them are important. So we need to keep an overall view of life and not draw any clear-cut lines regarding what defines life. Otherwise, we can get bogged down into one very narrow-minded view.
Yeah, I agree, and I still think that there are certain researchers out there who are making that mistake, just thinking purely in terms of the genetic component, especially. But I like Tibor Ganti's chematon model as like a proxy. There you have metabolism, compartmentation, and replication, and I think those are good, 3 good proxies, or pillars, as some people call them, to understand what we mean by life.
Right. All right. I'll play another section. I think I will play a little bit more. Otherwise, we won't ever get through this whole video. So I will try to minimize the commentary and maximize the amount I will play Eric Smith. All right.
Practical problem that is driving the set of questions. I want to know, what do we aspire to in a theory of the origin of life when, if we're realistic, our knowledge is overwhelmingly a lack of knowledge, huge gaps. Right now, what we settle for are often scenarios. These are descriptions of imagined histories. And the problem with scenario type reasoning is that it's very fragile when most of the links you would need to connect a chain, you don't have ideas of any quality for, or sometimes, most cases, any ideas at all. So
maybe I can speed up the video, like not by too much, but just in order to speed up a little bit. Let's see here. 1.25. I think it will help a bit. Not too much.
What we want is a way to be able to reason based on the things that we think we have better reason to believe that is robust against the presence of all of these gaps of context. In other words, we would like a science of the origin of life to look more like any other mature systematic science in a field that works. So how can we go about that? Oops, we've frozen out the answer. So I will tell you while we get the slide to start switching again. One answer to this, not meant to be the only possible answer, is that instead of looking for scenarios, we can try to identify sequences of dependence or of directionality. Do we go now? No, still not.
Oh, can you try now?
I can? Okay, yeah, now we're back. Okay, very good. So we can look for dependencies or directionalities that we can derive, even if we don't know what the interval between them was or what the mechanism was for them. And we're looking for those that can be done, or about which we can draw conclusions in the absence of much knowledge about mechanism. So let me give you an example of what I mean by this. Very well-known example. In 1961, at the first meeting to discuss the possibility for extraterrestrial intelligence, Frank Drake, to try to tame a conversation that he knew could readily get out of hand, tried to break the problem down into a sequence of different contingencies that one could identify, even if one could not calculate the likelihood that each one had within the one above it. So, for example, in order to have a civilization, you first have to have stars form, they have to have planets, the planets must be habitable, then they do or don't ever form biospheres. In the biospheres, there is or is not intelligence, it is or is not technological, and then it does or doesn't survive. So the reason the Drake equation works is that behind it, there's a kind of, oh no, wrong one. Now I've given away the punchlines. So there's a Venn diagram, and that exists because the final condition is actually a joint condition of all of the previous, which means that the number or the probability of finals can be written as a chain of these conditional probabilities. And even if we don't know what the values are, we gain a lot from understanding the containment relation of the Venn diagram. Now, an area where a hierarchy of this kind has been incredibly important to our theory, even as we have had to adjust our understanding of mechanisms, is of course the 20th century theory of the hierarchy of matter. So very early on, and because we have the good fortune to live in a universe with a big bang, advancing time is also advancing cooling. So very early on, you have a quark gluon plasma where you have independent action of symmetry groups on quarks and antiquarks. You cool a little bit, that symmetry is broken so that you now have only joint transformations. And out of that transformation, out of that breaking of symmetry, you get heavy baryons and lighter mesons. But at the first temperature, still, you're in a plasma of those free particles. If you cool a little bit more, then the exchange of the mesons between the baryons can condense them out, and you wind up with nuclei. But of course, you're still in an electromagnetic plasma with respect to the electrons that are not confined. You cool a little bit more, that can condense, and you form neutral atoms. And then from the atoms, you get molecules and condensed matter and so forth. So this theory exists because condensed matter.
This is of course very. This is of course in the realm of physics. But I think if I understand what he's saying here, is that you have different levels of organization, and in order to get higher levels of organization, something about the building blocks needs to be frozen in place in order to build higher order structures. And this is a relevance to the origin of life, where some elements at lower levels get frozen out, and then when they are frozen out, that allows for a new emergent order of organization later on, which I will also talk about in a few notes that I've prepared later on. So, do you have anything to say at this point, Drayce?
It kind of reminds me a little bit too of Eugene Koonin has his whole thing where he compares it to inflationary cosmology, and you see it like through the gradual cooling process, going through these phase transitions, like the gradual emergence of matter and galaxies and stars and planets and so on as the universe cools. And a similar kind of process is going on here with like the emergence of these structures.
Right.
Matter and so forth. So this theory exists because of another dependency relation, which is the chain rule for entropy. This is not the same kind of a containment relation as the Drake equation, but it is still a kind of containment relation in state space. So here, I'm not going to try to draw the equation for the entire sequence, but between any two stages in this, we have an important relation that to know a full configuration, we need to know its small scale degrees of freedom that are at higher energy, and its larger scale degrees of freedom. We need to know how that state space can be populated by the system. But of course, we can always write that population as a conditional entropy of the coarser degrees of freedom in the part of the state space that's occupied, plus the entropy of the finer scale degrees of freedom. And it is the presence of phase transitions that cause this entropy to be reduced and the part of the state space that you occupied to collapse into some corner that will then change the conditional entropy for subsequent organization. So in informational terms, phase transitions are two things that we're going to care about. First, they're thresholds, and second, they're scaffolds. So each time a phase transition causes you to fall into a corner of the state space and lose the ability to explore in some of the small scale degrees of freedom, the entropy of searching those degrees of freedom is no longer problematic for any subsequent exploration. So I got ahead of myself. First, it's a threshold. For most temperatures, nothing changes about the organization of the system. You hit a particular temperature, you reorganize. That's where the phase transition is. After that, nothing else happens for a while. So there's a threshold where either you form order or you don't form it. If you form it, then the entropy that's removed in the high energy scale is no longer affecting the problem of exploration for order at the lower scale. But here's where it's a scaffold. The thing that formed from the first phase transition is what we think of or experience phenomenologically as building blocks. And the subsequent organization is limited to the assembly of those building blocks.
It's the same thing that I mentioned before, like at high energy scales, you have these particles, but at some point, some of the particles become frozen into some configurations. And when they are, like as you said, that there is the entropy gets thrown out in a way, if I put it correctly in correct terms. But basically, when that state gets frozen out, then you have building blocks for higher order matter, basically. That's that's how I understand what he's saying right here.
And there's also like what he's talking about here in terms of reducing degrees of freedom, you know, sticking it over into a corner somewhere. That's relevant to the water paradox. Like everybody always, all these people, especially in the RNA world camp, saying how it's basically impossible to have these condensation reactions take place in water because it's thermodynamically unfavorable. But if you the way that people like Diemer overcome that problem, they do it through evaporation, which reduces degrees of freedom. But you can do it in an environment that is aqueous. You could do it in a vent environment where you have confinement that also reduces degrees of freedom. Yeah, and there are other things too, like silica gels and all sorts of other stuff that will facilitate this kind of chemistry.
And it's also interesting is that regarding the ribosome, like the very core of the large subunit is called the PTC, the peptidyl transferase center, which is the active site that forms the peptide bonds of proteins. And what is interesting is that the catalysis is not very stereotypical of most catalysis. It's more described as an entropy trap, where once you have these residues stuck in this small space, the very fact that it reduces the degrees of freedom and also excludes water molecules, that alone increases the rate at which these peptide bonds form. Basically, it's a very interesting way how the very chemistry of this of the ribosome works in a similar way that you are describing how water is excluded from the system, even though it's in the whole system is in aqueous solution. There are still some parts of it that can still make the system or make something dry in a way, and that drives the reaction forward.
Yep, serpentinization does that. Intercalation into clays with minor amounts of water. And all of this has been experimentally shown by now. Well, actually, not so much for serpentinization. That's more theoretical still. But in terms of it occurring within clays that have water content or occurring under nano confinement, this has been shown. And so there are a lot of these guys like Demeter Kunef asked me to watch David Catling's recent presentation where they're just repeating this mantra that this is not going to happen without going through these cycles of complete dryness. But that's not true. There are multiple different ways that you could facilitate this to happen, even in the presence of water.
And this is also like I have two main problems with the argument that it can only happen in complete dryness, and one of them is life doesn't need to be completely dry to drive these conditions. Like it's still in solution, and the way it drives these in a way that it drives these reactions, even though it's in solutions, mainly by coupling it with the disequilibrium between ATP and ADP and a phosphate. Like by letting ATP to become ADP and a phosphate that goes off, by coupling that reaction with dehydration reaction, it can still drive dehydration reactions even though it's thermodynamically unfavorable. But because it's coupled with the phosphorylation, that is or dephosphorylation, that makes the whole system favorable.
Yep, there's this paper. It's entitled "Generation of Long RNA Chains in Water," where they are using reactive phosphate of some kind. I can't remember which one it is, amidophosphate or trimetaphosphate, one of those, and they're able to generate long chains even in an aqueous solution. And there's another thing here too, but it's slipping my mind, so maybe it'll come up again in a second. But for now, that's that for me.
I also have seen a talk by Eric Smith again, but it's more of a personal back and forth between him and another person off camera. It's like about 4 hours, I think. But at one point, Eric Smith also mentions this thing about why people seem to go for dry or wet dry cycles, and he describes that as chasing equilibrium as a solution to the disequilibrium issue. So and he has something to say about it as well. It's the same way as I propose. Like Eric Smith notes how life thrives thermodynamically unfavorable condition by coupling it with a disequilibrium between ATP and ADP, and that is a very different solution than what, for example, Diemer, I think he's called, the wet to dry cycle guy. He's Diemer, right? Or Diemer. Oh, Diemer, yeah, like,
both of them.
Yeah. So of course, Eric Smith doesn't exclude the possibility that wet and dry cycles could have contributed, but he still thinks that you still need to explain why at some point wet dry cycles gave way to the pattern that we actually see in life with ATP driving these reactions. And maybe that organization with ATP has existed for a very long time without wet dry life cycles. Yeah, and that's one thing.
Yeah, and another, I guess the last thing here from me, I remember from before now, other mechanisms that cells use, such as molecular crowding. So like an example of this in terms of abiotic process that people like Kuhn and others talk about, you have these kinds of concentrating mechanisms, and when you concentrate them highly enough, Lane talks about the same idea where the thermodynamics switch, where you were originally favoring hydrolysis and now you're favoring polymerization because of concentration or crowding, and likewise within the ribosome, providing protection from hydrolysis within the ribosome as it goes about the function of peptide bond formation.
Right. Let's keep going on unless we can't go to the video, but I will play more. Yeah.
So this is how we have a sort of an successive elimination of conditional entropy that underlies our whole theory of the hierarchy of matter. So do those ideas apply to the living state? And if they do, how do we need to change them in the details from the things that we know how to do in high energy physics? Well, first, life is inherently dynamical. It's not just frozen states, but it's frozen processes. That means that the entropy we care about is the continually encroaching sequence of fluctuations. Their ongoing elimination is what we experience as dynamical control. We have a theory of dynamical control, now more than half a century developed, called control theory, and it allows us to quantify what any controller can or can't achieve. So when we see in the world an attained state of order, that must imply that for every controller that's eliminating the incursion of entropy, the context for the controlled system must have placed it someplace where the controller is actually within its capacity to achieve order. Where we get dependence and directionality is by looking at all the different ways we can assemble contexts and control feedbacks, such that there's always an adequate context to place each controller within the domain where it can function. And again, phenomenologically, we experience that organization of contexts and controllers such that the contexts provide paths of least resistance, and then the controllers do what it's easiest to do rather than doing something much harder somewhere else. So what would a context or a path of least resistance look like? For now more than 60 years, as we've come to understand more about the low-level building blocks of metabolism, it has been asserted by more and more people in more and more ways that the deep core metabolic reactions of life, which are universal across the biosphere and apparently go back to its beginning, certainly look like something that was prefigured in what it was easiest to do in chemistry. So the content here is, it turns out that everything in the biosphere is synthesized by a pathway that starts from one of 11 molecules, actually one of 4 or 5 among these 11. And it happens that there's a pathway that goes through the 11 that is a metabolic pathway. If we look at where there's variation in the biosphere, we find that there's variation in the ways you can fix carbon. I won't take the time to explain to you this diagrammatic, but the publications explain more. It's just a chemical reaction diagram in a sort of a computer-friendly notation. But of the carbon fixation pathways, they all must be self-amplifying, like Leo Szilard's famous chain reaction. And it turns out that 4 of the 6 known pathways, very possibly the only 6 that exist, are all either part of this same core, overlapping with it, or tightly linked to it. So it has been proposed that through a combination of their chemistry, their centrality, a variety of remarkable properties of this, these look like paths of least resistance that should have come up in geochemistry and been the easiest thing to do, so that when higher levels like molecular control came into existence, they reinforced what was easiest rather than replacing it.
And this is also a very interesting talk that Nick Lane often talks about regarding the citric acid cycle, or the TCA pathway. It's specifically the TCA cycle. This one has a very interesting feature where, if you, or in us, it goes one direction where you consume, for example, sugars and put in pyruvate, which gets broken down into carbon dioxide, and then from this citric acid cycle you produce I think it's NADH, which transports electrons to the electron transport chains, where you can get the ATP synthase going and such. The whole story of how ATP gets made, but in other life, and perhaps and probably in the oldest life forms, the citric acid cycle goes predominantly in the other direction, where you draw up CO2 and you start producing these molecules in the opposite direction. And which is very interesting. Like at this point, when you produce citrate, it splits into these two molecules, acetate and how do you pronounce it? Oxaloacetate.
Okay. Yep.
And so if you continue this cycle over and over again, it becomes exponential. First you have one molecule, then becomes 2, 4, 8, and etc. It's a self-amplifying feedback loop, which is the characteristic of a autocatalytic network, basically.
And this is basically what you need when we're talking about this core metabolism for the production of amino acids, lipids, carbohydrates, nucleobases. This is something where, again, people are saying that hydrothermal vents don't offer you the chemistry. Yes, they do. You get to, for example, using iron, you get 9 out of the 11 of these. The only 2 that you're actually missing are citrate and oxalosuccinic acid in this cycle, but you get oxaloacetate using iron. And then from there, if you have a system that has zinc, like hydrothermal vents do, you are able to transaminate to aspartate, and then aspartate to dihydroorotate to orotate. And then if you have copper, it's not just orotate, you also automatically get your uracil. So you get precursors for your nucleobases and nucleobases with the metals in the system, just operating off of what you get from the protometabolism caused by iron, giving you all of these products of this core metabolism.
Right. And it's also interesting that many of these, I think 3 of them, are like the carbon skeletons of amino acids, like also the very early amino acids, probably like the special ones, for example the aromatic amino acids and histidine, especially like those amino acids that we need to consume because it's very expensive for us to synthesize. So us heterotrophs can decide like, oh, we don't make it ourselves, we just eat other organisms that will produce it for us. But the simpler amino acids are direct or a few steps away from being direct reductive emanation of these carbon skeletons of the citric acid cycle component. Yeah.
Yep. Yeah, you switch them around, they become like interconvertible.
Mm-hmm.
Yep.
So it's very interesting that at the center of core metabolism you have only carbon, oxygen, and hydrogen, of course. But then just one step away or a few steps away from this cycle, you introduce nitrogen, and at some point you also introduce sulfur as well into the components. But of course, I will play. If you have anything else to say, Drayce, or not?
No, let's continue.
I'll continue.
That's, of course, an experimental hypothesis, and its testing is still underway.
This is what you were referring to just a few moments ago with when you introduce different metals, you can drive these reactions in the citric acid cycle, right? In this figure.
Yep. That's Varma, Moran, Muchowska. They're a really great team. Harpreet Kaur is another one who's on the team. All of these are people that should be watched closely for the work that they're doing now.
Yeah, and it's also very interesting. Like even though these reactions look very complicated, some reactions are just kind of duplicated. Like one reaction is very similar to another reaction. It's basically you only need to catalyze very similar reactions to get the whole system going. Basically, that's the beauty. That's the kind of the beauty of this whole core metabolism aspect. But I will let Eric.
That's of course an experimental hypothesis, and its testing is still underway. So for a perspective on this that I think treats the most difficult ideas in the most even-handed but also conceptually sophisticated level, which we often don't find, I can refer you to this wonderful review by Kamala Muchowska, Srijit Varma, and Joseph Moran. Much of the most exciting work that they report on actually is done by these investigators and their collaborators at University of Strasbourg. And this is one of the plots from their review paper and papers before it, in very simple systems that are meant to capture some of what should be going on in early planetary surfaces. They do, in fact, create multiple carbon molecules that pass through every one of these drawn molecules is, in fact, a biomolecule in that low-level system that I showed you. And in fact, with very simple catalysts, soluble iron and zinc in certain oxidation states, they even can reproduce segments of the biological pathways as they are used. Now, the organization of this experimental result is not the same as that of biochemistry, and we don't yet understand what it is about the way we're asking the question that causes us to not be able to learn more and decide this in a better way. But I think it's fair to say at this point that enough good work has been done that it doesn't look crazy to say that a lot of life was not brought into existence in the hierarchy, but rather is prefigured in very simple low-level laws that come originally from the periodic table. So, of course,
now it becomes a very interesting part of this talk that that prompted me to discuss this, because I find it very eye-opening as well. But I will let this continue on unless you have anything else up until this point, thanks.
Nothing currently.
Oh, I will let him talk, and it's very interesting. So keep note.
Life is not made of complete sentences, it's made of being in a hurry and speaking in shorthand. And so when these questions are expressed in shorthand, they're often put forth as a kind of an unfortunate dichotomy between whether metabolism or genes were first in the origin of life. This putative debate, okay, for an approximation, is really about the organization and the capacity of information flows. In the debate, as cartooned, the metabolism-first point of view is the one I was telling you earlier about conditional entropies. The idea being that the selection of metabolism was done before you have higher order molecular systems. The alternative point of view is one that really takes a kind of a brute Darwinism direct from modern biology and tries to impose it right from the beginning and says control flow from macromolecules is the reason anything exists. An interesting thing which I want to get to is that while the two sides have very different approaches to information organization, something that they both tend to do, I think more through oversight than anything else, is they tend to conflate the macromolecular world as being folded in physical structure and also coded with a code achieved by selection. That's where I want to say something to you that I think will be new today, claim that those two should not be conflated, they should be separated. That in fact, there's at least a three-level Venn diagram. The metabolism-first idea is the one that I already showed you, but we should in fact separate folded from coded matter as categories. So why?
So what you're basically saying is in reference to the talk at the beginning about the condensed matter at different stages. Like when you first say you have many degrees of freedom with the building blocks, but then some of them get frozen out into larger structures, and then they form the building blocks for later organizations. And what he's basically saying is that first, protometabolism gives you some structure that gets that has a very low degrees of freedom. Of course, still some leeway, of course, but then that gives rise to matter that is folded before you get coding that codes for these structures. So that's very interesting. You're muted, Drayce, so if you have anything else to say, you feel unmuted.
No, nothing to add. This is this is getting back into emergentism, but it is interesting here making this distinction between folded and coding, and like the kind of implications this has for a lot of people who are arguing for this intimate structure-function relationship, that's inextricably intertwined and you can't separate them. So I think this has some ramifications here. We'll see.
Why, in a world where people can disagree on many other things, would they make this conflation in this way? I think it comes from the fundamental difficulty of figuring out how to situate the central dogma for control flow that we know from biological evolution in a larger physical theory. So the central dogma was articulated by Francis Crick to try to capture the role of genetic and catalytic systems in a Darwinian paradigm for evolution by natural selection. The idea of the dogma is that variation enters when DNA is replicated, or in viral life cycles, RNA. There is control flow where everything is a template for everything else, where DNA can make RNA, RNA encodes protein catalysts, which then catalyze the reactions, and the molecular substrate, which is organized by the catalyst, is then the material out of which all this is made. So the dogma binds all of these steps into an unbreakable ring of just the nastiest chicken egg dependencies, because that's not super complicated, but that really is. Now, the discovery in the early 1980s that RNA could be catalytic created a seismic shift in the field, and everyone went running in the same direction because they thought, aha, we can get away from the hardest part of this, which is the RNA-peptide relation. But they kept one commitment of the central dogma, which was the downward control flow. Variation is introduced a la Darwin in an a priori unbiased fashion. We have to wait until it gets to the expression of phenotype to decide whether it was good or bad, which means we need all of this material to generate phenotype. It is important in this that folding, which actually makes usable structures, has no agency. It presents sequences for selection, but it does not select the sequences within this Darwinian control loop that has been taken from modern life and sort of imposed on early chemistry. The discovery of RNA catalysis may allow us to shorten the loop, but it does not tell us whether it should be broken. So the next question, should we, and if so, can we, break this ring at folding?
So what it's basically saying is that there is like a chicken and egg paradox, quote unquote, because it's not really a paradox. It's just like it's a very a solution to it. It's like what we call a paradox is basically a problem where a solution is difficult to intuitively impose. That's how I see what a paradox means. So it doesn't preclude, like a paradox in literal sense, precludes a solution, but not in this case, in my opinion. And what he is basically saying is that perhaps this chicken and egg relationship between in this very complicated system is the difference between folding and coding. Perhaps you have structures that folded before they were encoded by a genetic system. That's what he is saying, if I'm not mistaken.
And in terms of the whole thing about this being a paradox, it's a paradox to a bunch of people who are operating as vacuum heads, where they're just considering their one preferred approach, whether it's like RNA or lipids or whatever, and only RNA, only lipids, and just considering these things in a total vacuum without actually considering like the full context of the environment and the diverse chemistry that it's driving. Because if you're synthesizing amino acids into peptides and nucleobases and sugars and phosphates and nucleotides in the same environment, there's no paradox here. They're both present.
Right. I think this also has to do with Dimitar Kunev's challenge. Right? Like I think in his talk at one point he proposes a challenge, and I think this has some relevancy to his challenge. Like, oh, you need to explain. I think it's the part where he says you need to explain structure and function, right? And this on the answer what Eric Smith is basically giving is structure came before function, basically.
Yeah, I'd be interested to get Dimitar's response to what Eric Smith says here. It does seem like he's trying to meet Dimitar's challenge, not consciously, of course, just by happenstance.
Yeah. Right, exactly.
So let me give you two approaches to the answer. This one's just kind of evocative. Think about states of matter. Think about condensed matter, solids. Certainly, we could make condensed matter by putting every atom in its place with an atomic force microscope that we paid several $1 million to purchase. But do we really think that condensed matter only exists by virtue of the fact that it could be assembled with an AFM? No. other way around. And yet we do think that way about folded proteins, folded RNA. We contextualize them only as consequences of sequences which are produced by a selection model. So
it is really interesting. Like this was really an eye opener. Like as somebody who has of course learned molecular biology from since high school, I always imagine proteins as something that is the direct product of an encoding system. Like, oh, every amino acid in protein is specified by this complicated chain of transcription and translation from DNA to RNA by the ribosome. And basically every amino acid is specified beforehand. But what he is saying is that maybe it's not like this from the beginning, at least. Maybe you can have something like a protein, maybe not, of course, modern proteins probably did not exist before genetics, but maybe you can have at least some form of structure or folding with RNA and polypeptides before they were encoded by a genetic system. That's it.
Yeah.
All right.
So do we really mean that, or are we just being sort of thoughtless and hurried? But let me give you another argument that I think is better. This one from control theory. Leslie Valiant wrote a wonderful sort of early stage book and a paper leading up to it on what he calls PAC, or probably approximately correct learning. Valiant starts with an observation that we all know, which is that selection is the same as Bayesian filtering, which is the same as reinforcement learning. He goes on to say, or to conjecture, that the learnable concepts are a bounded category, kind of like the computable functions are a bounded category. And because selection or reinforcement learning is not a strong algorithm, it's actually quite a bounded category. So there's a lot that can't be learned. So the way we would apply this is to say, if you want to say that something came into existence plausibly in a random system, then you need the variety of your controller, to use a Ross-Ashby term from the 1950s, the diversity of system states that it can sense, discriminate, and act on. You need that variety to be within the controller you can actually make. So what is it that reduces the required or the requisite variety of controllers? It is for the controlled systems to be, for instance, constrained a priori, or to have self-governing mean regression over some of their degrees of freedom, so those don't have to be regulated from the top, or concurrently acting processes so that multiple controllers are acting collaboratively. So where would prior constraints come from in folded matter? Well, some of them can come from the metabolism that we already talked about. So really terrific review paper by Ronells et al. Senior author is Lauren Williams, and I'm going to show you several papers from Lauren's group. Says that, in fact, the monomers that ever form...
Lauren Williams is also a very interesting guy in the origin of life research. He mainly talks about the ribosome, but it's very interesting papers that he has on that, and his team has published.
Yeah, I really like him, and he's another one who's been really pushing like an RNA peptide partnership. I think that's important, and I think that's going to be like, the future. One crux, or like the future of origin of life research, and that, like another question here to like situate like folding, because folding also protects against hydrolysis. So like there could be selection for folding that's like non-Darwinian selection for folding prior to any kind of coding and stuff like that, simply because of like selection for things like degradation from water and so on.
It's very like if you randomly polymerize things, then those that happen to fold a bit, they will remain longer than the ones that the ones that don't fold are going to become monomers again, which basically bias everything to become more folding over time, which is interesting, yeah.
Yeah.
Form folded structure are a quite remarkable subset. They're the polypeptides, which make beta sheets and alpha helices. They're the polynucleotides that we know of in RNA and DNA. And then they're polysaccharides that make sheet-like starches. What they all have in common is that the medium strength bonds, the hydrogen bonds, are present in sign and in number, so that when these are arranged in space, every bond that exists to be saturated is saturated by another bond in the complement molecule. So now you have a very complicated mixture of organic synthesis, counting, positioning in space, the number theory of packing things, And the things that really fold have to satisfy all of these at the same time. So you have a lot of information in what your synthetic system provides before we've ever talked about how folding happens. When we do talk about how folding happens, that turns out to be a hierarchical entropy rejection funnel in its own little world of rules, kind of like the chain rule in microcosm that we talked about earlier. So these rules are governed largely by the hierarchies of energy scales. If I take monomers and they're free in solution, at some dilution, they won't aggregate because there's too much entropy. If I bond them in a chain with a strong covalent bond, that takes out enough entropy that now the favored state within the molecule can be to aggregate, but not necessarily to make a fold. The next thing that acts slightly lower in energy, but first because it's cooperative, is that the sequences that I make things out of can be more or less soluble. And if they're properly arranged, the insoluble ones in water group together so that they minimize the exclusion energy of water. That will collapse you to what's called a molten globule phase on the way to a fold. And then if the residues are right, the monomers are right, the hydrogen bonds are available, then that molten globule can anneal under the weak force of the hydrogen bonds. And I'm giving you each of these in kilojoules per mole relative to KT. So you can see we're getting down pretty close to the thermal fluctuation energy. And then last of all, so the annealing gives you your secondary structures, beta sheets, alpha helices. And then only after all that's done do the weakest of all, the van der Waals forces, act collectively as a surface to fold you into the tertiary structure that kind of locks it all in place.
And it's a very, there's also like one thing that we learned in the last decades about protein folding. Like when I was in college, I learned about what's called the Levinthal's paradox, where a protein of a decent size, like a 100, let's say 100 or 200 amino acids, it has a practically infinite way to be organized, but yet proteins they manage to fold into a stable structure nearly instantaneously. So, but how can it find the right solution in a practically infinite number of different configurations? And what this is showing is that you have a step-by-step, you have many different forces that will step-by-step find the right solution very quickly, just because of the it rejects the entropy in each step. That's really interesting.
Pretty awesome.
Yeah, and then I think it also has to do with I think he talks about this later, but this also plays in why folding is probably independent of encoding, because you can have folding without being encoded necessarily. It's, of course, encoding ensures that you have the right sequence, but in order to find the right sequence, it doesn't necessarily take encoding. But the system of encoding does require you have very sophisticated folding structure. So it's another argument for why folding came before and coding became later.
So now we can.
Did you have anything to say, Drayce?
I wanted to ask your personal opinion on the argument that structure and function are intertwined and can't be separated. Do you think that is, because this is like a criticism that I've made before, do you feel that suffers from the same kind of reasoning as like irreducible complexity arguments?
I think I have like I think I know what you are getting at, but I think the objection against irreducible complexity is a little bit different than assuming structure and function are inseparable. I think that's what I'm saying.
Okay.
Like, I think irreducible complexity assumes that, for example, like there are many versions of irreducible complexity. Like some versions don't allow for functions to change, but if you allow for function to change, then you can build up an irreducible complex system by changing functions along the way when you build up complexity. And another way that defeats the irreducible complexity argument is assuming that you can only add one block at a time. Like if you have block A, then A plus B, then A B plus C, and A B C plus D. That linear way of complexity building rejects other possibilities where you can have two or model independent systems. Like for example, you have one system ABC and another system B or ABC, and another system D, E, F, and then later they can combine to form a new structure, even though you can separate them that way out, which doesn't allow for a linear way of complexity building. And another way that you can defeat the complexity argument is basically you have something called neutral constructive evolution, where you have one complex that performs one function, but then you have a change, a neutral change, where you add more complexity, but then later on something happens where that additional complexity becomes necessary for the system to function. So you can have an evolutionary ratchet where you build more complexity even without being adaptive as well. That's a different arguments when it comes to structure and function, I think. So, I know, do you think there are similar objections to the structure and function argument?
I think the way that you responded there is fair. What I have in mind is more that that on a micro level, there can be situations going on that have absolutely nothing to do with the function that you want to account for. So for example, here we have a dynamic that leads to folding that is independent of function. But yet, over the course of time, the micro level behavior coalesces into something that works together reciprocally between these various different things that are going on to produce some higher level order, and that's kind of what I was getting at there. But I think your response is fair. Yeah.
I think my main argument against the structure function of treating them as inseparable is the same way that how you argue. Like you argue, as you say, that the way these structures get built physically is independent on it's blind when it comes to function. So assuming that function and structure are the same thing it's basically, I think it's a fallacious teleological thinking, right? Like it's assuming that, for example, if I pick up a rock on the road or in the wild, then I assume that it somehow has a function to scratch my back, to bring up a random example. I think it makes the same mistake.
Understood. Yep. I understand what you're getting at with the whole teleology thing. I have a slightly different approach on that, but it's for another time. It's actually not as related to this as it should be if I'm going to discuss it here. So I'll shut up about that for the time being.
Oh, yeah, sure. But if you want to bring it up again later on, then we can do if we have time. No worries.
We can ask, how much information do you need to make use of this funnel for folding? Which brings us to 15 years of remarkable work done by Michael Hecht to provide a partial answer to that question. Hecht's system says, look at the arrangement of an alpha helix. This is looked at end on the spiral, and say, what periodicity could I put just hydrophilic, hydrophobic residues in so that the hydrophobic ones can all share this kind of folded center and the hydrophilic ones can face out into water? And can I make real folds that way? The answer is yes, but the periodicity is this. For beta sheets, it's actually a little bit simpler because residues point up or down, and in certain contexts, which I don't have time to go into detail on, with almost complete freedom in which hydrophobic or hydrophilic amino acid it is, as long as you put them in the right place, you can make amyloid-like beta turns. So what Hecht has done is taken the point of view that I can just have broad equivalence classes of what the chemistry of my amino acids is. I have a relatively local condition for what the correlation length is, and out of that I can get folds, but there's still a fair amount of information that he needs to provide because he needs that local condition to be repeated. So it's a kind of a purification argument, a little bit like what we have when we say that we're using the right monomers and not the wrong ones.
I guess if I understand it correctly, like he's getting at like how much specification do you need to get folds? Like even though right now we have 20 amino acids, and arguably the early amino acids were at least I think the oldest ones are 4, but I think a decent amount is like there were 16 at first, and then four special ones were added later on, if I'm not mistaken. Drayce, do you know anything about which amino acids came, or what the number of the original amino acids were?
I would be a little bit of a heterodox on that one, because the way that I see some researchers talking about what counts as prebiotic amino acid and like the sequence by which they were added to the code, I think some recent research is going to upend a lot of that standard narrative. In some articles, in some articles you'll read that cysteine is not prebiotic, but since just before 2020, they've had success synthesizing cysteine abiotically. Same thing with tryptophan. That has been shown to be abiotically produced at hydrothermal vents even today. And so there are examples now where we thought certain amino acids were not prebiotic, but in fact probably were. So I think that's probably going to change a little bit over time.
Thank you. I think I was mistaken in assuming that the amino acids that were added to the genetic code later on did not exist before. They certainly existed before the genetic code, of course, but that was my mistake to conflate the genetic code with which amino acids are older. They are necessarily older or younger relative to the genetic code. But yeah, that's my bad. But I think regarding this piece, I think he's talking about like you don't necessarily need to have a highly specific sequence of amino acids. Like he says, at this position you need one hydrophobic one. Like it doesn't matter which one, as long as hydrophobic, and then this one hydrophilic and this one hydrophobic. So you need less specificity when it comes to these simple folds. That's what I'm getting at from this portion.
That makes sense. Yeah.
Yeah.
But you may be able to do better. It's so great to talk to an audience with general relativists in it, because I can tell you, or I don't need to make the argument to you, that the coordinates that we use may not be the geometry that the system cares about. So my coordinates in space for an alpha helix are the straight Euclidean lines, and in those coordinates, it's 3.6 residues around a turn to make an alpha helix. And that's terrible because it's not an integer and it's not small. And that's why Hecht has this very complicated periodicity that he needs to maintain, like 6 or more. But if we look at the actual cores of small molecules, this beautiful thing is called a Tim barrel. It's an 8-fold repeat of alpha helices, red, beta sheet, yellow. And on one of these, I took out the cartoon so I could show you the backbone, and then I pulled that up here. This little thing is kind of the hydrophobic core of the core. And we find this motif over and over in all the oldest and all the simplest folds. But the world that those carbons live in is a world of tetrahedra bound in chains, and that world twists. And in the twist, the natural periodicity in the helix is 4. And the natural periodicity of the beta strands that couple to it is 2 by 2. And I don't need to tell you that 4 is really neat because it's an integer and 4 is 2 times 2.
I mean, this part at first confused me, what he's talking about. But if I understand correctly, like if you look at an alpha helix, for example, he says that you need to maintain a periodicity of 3.6, which is very complicated. Like if you key it has a very high, you need a lot more information or specificity to maintain that periodicity to make an alpha helix. But when it comes to the simple cores of proteins, then you have this other periodicity where you have 4 with the alpha helixes and 2 by 2 with the beta sheets. So this short periodicity is very easy, much more easier to produce. I think that's what he's saying right here. That it takes less quote unquote information to produce this structure or this motif, basically. Yeah, I'm interpreting him pretty much the same way. Yeah, I think I'm understanding it correctly this way. Maybe I'm misunderstanding, but that's how I interpret this portion.
So, all of a sudden, the periodicity that worked for beta, if we can coordinate that twice, starts to become periodicity that looks relevant to getting the most molten core for one of the oldest folds. So, I think we can argue that making use of folds as a funnel is not vastly improbable in highly random libraries. But what would we need to say that this existed historically, or that it did important work that we can take off of the shoulders of a Darwinian loop? Well, we would need evidence placing it before translation, which is when coding is expressed. And we would also need at least a putative mechanism by which folding could actively select sequences and not just passively present them.
This is also an important thing that to show that folding was folding became before coding, where you like the translation, at least some form of translation is required to have an encoding sequence, right? Like you cannot produce an encoded protein without being able to translate your RNA sequences. So if you can show that folding came before translation, then it's a good start. And regarding the mechanism of basically what he's saying is that if you have a way to select sequences based on whether they can fold or not, then you have a plausible means to get folding without folding merely be a representative a representation of the encoded sequence. That's what I'm getting at. That's what I'm getting from this.
I concur.
All right.
So that's what I'll show you now. This is the ribosome. This is where everything that's hardest in biology is all brought to the height of tension. This is a significant fraction of 1 million atoms, RNA, peptides, metals, water, every atom in its place. I want to argue for you that it's also the place where we have the most empirical evidence for how we maybe untie some of this knot. So it's been known for several decades, including the work by Tom Steitz and collaborators, that the ribosomal peptides, the proteins that make up the ribosome, here what we have done is we've ghosted out the RNA so you can't see it, and you're just looking at the proteins in the small subunit and the large subunit that together make a kind of a ratchet that does your translation. And you can see that they're not the same everywhere. On the outside, they have complicated globular folds of the kind that we think of as high information. But as you go toward the inside, they're long and stringy, and in fact, the peptide backbone doesn't even touch itself. It just touches RNA. So there's clearly a change in the character of information. Hyman Hartman and Temple Smith, I don't know if they were the first, but certainly they were early ones to propose that this is not just a gradient in space, but it's also a gradient in time, that the simple was early and the complex came later. The thing that really cracked all of this open was the 2 Ångström crystal structure of the RNA and everything that we could learn from it. So in this wonderful paper, again by Steitz's group, they introduced what they call A minor interactions, which turn out to be important to stabilizing the 3-dimensional structure of ribosomes. The way this works is you have a helix like this gray one here, and then you have a new helix that comes in, the orange and the green one. And in the new helix, if you have a stack of 3 adenosines that can turn out, they can pack into the minor groove of the pre-existing helix. And it's a really nice tight thing. So it'll latch this green and yellow thing down onto the gray one. And if I look at the ribosome, these little red tiles, this is all the A minors. So they're everywhere. It's full of them. 5, okay. So this relation is inherently directional. Konstantin Bokov and Sergei Steinberg said, let's make a directed graph of all of the A minor interactions, and let's find out whether it's cyclic or acyclic. And it turns out it is acyclic, which means that there's a center and there's a surround.
It is also really interesting about the ribosome that apparently it's apparently made up of layers where as you can see here, some portion of the ribosome foundation of the other part that was added later on. At least it seems to be to tell this from this relationship between one side of this interaction and the other side of the other interaction. It's really interesting. I think he will say more about this later on. But yeah. Race, do you have anything to say, or like you're unmuted? So I see you are muted and you have something to say.
Just one word: onion.
Right, exactly.
So, in an extraordinary series of papers done within Lauren Williams's group, the first author in this is Anton Petrov, they have gone back, and for both the large and the small subunits, they have been able to take the secondary structure and completely decompose it into a set of helices that were there before and new helices and other structures that insert into them. And they have shown that, in fact, this whole history decomposes into a kind of onion with 6 phases.
There we go.
And I'll show you those in the next slide. But the important thing is,
feel-
layer by layer, the onion is a compact structure. It's stabilized by internal interactions, and it's suggested to fold autonomously. So, moreover, the structural dependency, which is sort of what you need to make and fold the thing, is recapitulated in the evolutionary branching tree of relatedness. The things that are old are also conserved. The things that are deep are also conserved and evidently old. The things that are outer are more variable and evidently newer. This is where they've shown that, in fact, the Volkow-Steinberg dependency can be resolved into 6 phases. Within a phase, you can't say who comes first and who comes second because they don't depend on each other, they're commutative. But across phases, everything in a later phase must have been preceded by everything in an earlier phase. And here's the real remarkable thing. The interface between the large subunit and the small subunit, which allow them to come together to make a ratchet that translates RNA into proteins, that's this yellow layer. These two things first met each other, and there was the translation system at the end of the green layer and the beginning of the yellow. So we have all this organization from folding at a time when we know that what it was doing was not translating. If we look at
this first piece of evidence that folding, you have folded structure of these ribosomal units before the whole ribosome was able to translate things. Because like without the interface between the large subunit and the small subunit, you cannot perform translation. At least not to the extent that the ribosome does today. Maybe the precursors of the small and large subunits were able to translate or perform something like translation, but definitely not to the same extent that we know it today. So what he's saying is that this points to folding having occurred before you have something that encodes the sequences to have these folded structures.
Look at the peptides that I showed you to start.
Anything to say?
I wanted to apologize. There's been a couple of times here where I preempted something that he was just about to discuss. I think I soaked in this lecture a little bit more than I thought I did. I guess this is actually the 3rd time I've watched this. It's a good lecture, but that's my bad. I keep there's a couple times I've introduced the topic before he starts to go into it. So my B.
I don't know. We are reviewing, so there is always we will always inevitably be talking about something that will be mentioned later on. So that's not too bad, of course.
It turns out that their gradient follows the phases of the RNA. It groups with them, and in the deepest, lowest, and oldest, they're long and stringy, and they get more complex. And we would think of as having more information conditions, with the beginning of translation beginning to make the complex globular ones possible as stable parts of the lineage. So in all of this literature, these are fine, careful scientists. They know what they mean when they talk about structural dependency. They can back up everything they say about evolutionary relatedness. They typically do not like to commit to a proposed dynamic that would have caused all of this. The reason being many dynamics are confounded, and so it would require a big speculative leap to propose one and not others. That's why I'm here to talk to you, because I'm not as careful. So among the confounded possibilities, to show you how bad it gets, could be that everything in the biosphere came from one molecule. We can't rule that out. Or it could be that it's Darwinism all the way down, which is kind of the RNA paradigm that I showed you before. But I think a better prospect for this is that processive folding was a kind of a kinetic filter that distilled sequences in attractors, a little bit like the composomes that Charlie mentioned from Doran Lancet and Daniel Segre. That the early branching effects that we see in the phylogeny have to do with the growth of the basins, and that the early large and small subunit functions were kind of just hitchhiking on these things that existed for the sake of folding, not for the sake of some downstream function. So what kind of a mechanism would do something like this? Well, something not very different from precipitation dissolution equilibria. Donna Blackman has done a lot of work on taking a mixed solution of small molecules with different handedness, different chirality, and showing that you can pump out that entropy of mixing just using the binding energy to form crystals that are chiral. Hans Kuhn, way back in the early days of sort of complex system and cybernetics reasoning, was arguing that if you put these binding on binding type transitions in a system that is driven away from equilibrium, you can break symmetry and you can cause the driving to make a version of what today we would call kinetic proofreading. So the conjecture is that in an early world, lineages come up basically only once you can survive, and that requires that you fold. So if you have a folded structure with loose ends, things that don't match the fold can be hydrolyzed off and you can try again. But if they fit, then they go in, just like fitting into a crystal, and then that becomes part of a stable structure. So folding can take on its own agency.
That is something that I mentioned before, like he's now proposing a more precise mechanism for why folding can be selected, even though you don't have anything similar to Darwinian evolution with the translation and transcription system basically in place. So what is basically seen is if you have random polymerization, and just by accident some fold better than others. The ones that fold better persist for longer, and they can and then you can have maybe more things that fold better, and then maybe you can add maybe some things that fold better survive long enough such that more structures can be added onto them just by more polymerization. Like it did, for example, like you can see my cursor, I think, right? Can you see my cursor?
Yes.
Like you have this, for example, you have this pre-folded structure, and then this failed to fold, so this gets preferentially hydrolyzed and cut off, and then maybe later you get more things that polymerize on it, and sometimes unfold, so it gets added onto the structure over and over and over again. Basically, it builds up. That's what I'm understanding from this part again. Yeah.
Yeah, prebiotic selection rears its ugly head again. That pesky concept that the creationists hate so much.
Mm-hmm. Right. I think like it's all like we all even some abiogenesis people get often bogged down into the specific Darwinian dynamic bit where you have individuals having variation and reproduction, and some get selected and some get weeded out. But what this shows is probably you can have selection without strict replication or without strict replication or strict Darwinian evolution. Basically, you can have selection based on what structures are more able to withstand degradation. Basically, that's what I am thinking about.
Yeah, and I really agree with that. I think it's just because a lot of people who have gone to work on this question, those of them who are evolutionary biologists, are just so used to thinking of selection in those terms, and they're less familiar with other potential driving forces like thermodynamic driving forces. And so the idea of prebiotic selection in a non-Darwinian, non-hereditary kind of way seems really foreign to them. I'm hopeful that more people will come to be comfortable with that kind of thinking, especially since in evolutionary biology, it's already recognized by a lot of people that selection is a multi-level phenomenon. So even within Darwinian selection, you have selection operating at different levels and stuff like that. So maybe they'll be able to understand that there's more than one way to understand selection and how it operates.
Right. I think it also, like Eric Smith in the previous slide, he mentions that functions were basically hitchhikers onto these folding. Basically, what he's saying, I think what he's saying is that this like folding were not like they didn't fold because of a particular function. They just fold because that's how things are more often survive than non-folded things. But then when they fold, just by accidental process, they may be able to perform some function that can feed back onto the cells. For example, if you have several things that fold and they interact in such a way that will increase the amount of polymerization or maybe preferential polymerization, then you can basically enter in a feedback loop where you begin to get something that resembles self-replication or maybe even encoding. That's what I'm thinking about right now.
All right, cool.
Yeah.
I am at the end of time, but I want to give you what I think the conjecture for this is, because I think it's really fun. We can ask, was the ribosome actually the first conveyor of lineages, the first thing that we would think of as a genome? We know that folding competence is a precondition to anything because you got to survive and not get dissolved. We know that if you have a strand that's folding competent, its dual strand is not generally folding competent, which means that that's going to be transient and gone. So the ancestor of the coding strand today has no status in this early world because it's not folding competent. The ribosome thus was not what it is today, a transcribed copy of a hereditary signal. Rather, it was the carrier of heredity. And the only thing that allowed its complement strand to ever become what we think of as the coding part of genome was, in fact, the emergence of translation that was not created by that genome. So our summary. Minimizing information requirements is a real principle that we can get from counting and that transcends many mechanisms and our lack of knowledge.
And this is like one last thing, what he's saying right here about the ribosome being the first genome that wasn't encoded by a different sequence, like right now in our own genome. We have in our genome we have ribosomal genes, right? Like that encode the ribosomal RNA and also ribosomal proteins. But what he's saying is that early on, the ribosome itself was the sequence that was that encoded itself basically. So it was its own genome, and it reminds me of several papers. I don't know remember which, but I think I've seen several papers where it says that the ribosome, the RNA sequence of the ribosomal RNA, they contain sequences that are able to encode things like ribosomal proteins and also things of the even the amino acetyl tRNA synthetases, the things that charge the transfer RNA with amino acids. It seems pretty interesting that yes, the ribosome could contain this information basically to encode these things as a genome in of itself.
Yeah, and that makes a lot of sense. You know, and I think here it's pretty compatible with what Demeter has going on with his bridge peptide. You'd expect, at least I think it's harmonious with what Eric Smith is doing here with ribosome as the first genome. And you could still have Demeter's bridge peptide ultimately facilitating the first like replicative relationship between peptides and RNA.
Mm-hmm. Right.
He uses it to. He uses it like basically his bridge peptide is the precursor for the aminoacyl tRNA synthetases, and then Chatterjee builds on that where he accepts Demeter's bridge peptide as like the ancestral AARS, and then ribozymes, variations on ribozymes leading to like your mRNA, tRNA, ribosomal RNA, and so on.
Right.
Depend on the materials we're talking about and their state spaces, so we can make specific hypotheses. I will argue there should be key, irreplaceable transitions that are the information gateways, and we can reason about these from laws. And in particular, that there are 3 of them in this talk: metabolism, folded matter, coded matter, in that order. And if you think that this is a plausible argument, I think it entails that the ribosome is our best candidate for the first genome. So I want to acknowledge many people who have worked very hard to help me learn to think about this stuff, though anything nutty I say is my fault and not theirs, especially Anton Petrov, who's given me extraordinary time, but the entire Williams group and all of my collaborators here, and collaborators at LC, who are also experts in folding and random libraries, the institutions that have kept me alive materially and cognitively, and these are better versions of the citations. Thank you all.
There are also a few interesting
so much,
questions that
Eric.
were
This was
asked by the
fascinating.
audience. So I will let it play out.
Yes. Microphone is on its way. I didn't see who was first.
Yeah, you talked about the ribosome being the first genome or something, and then, but earlier you said that the 2 pieces, the large, the LSU and the small unit, had features that you said was not meant for translation, as if you could figure out what it was doing before they what these 2 pieces were doing before they became part of the ribosome. What are the speculations about what that functionality was? That's important if you want to say that it was the first genome. I assume you mean that not the ribosome, but the most fundamental conserved parts of each of those subunits are what was there before the ribosome.
And after it became a translation apparatus too, through that whole transition. But yeah, terrific question. It is believed by a great many people that what the large subunit was a general purpose condensation catalyst. Polyesters, polypeptides, ester-amide exchange, these kinds of chemistry that are ubiquitous in the deepest and oldest parts of everything having to do with nucleotide and polymer formation. The small subunit seems to be believed to be related to RNA coordination, possibly helping template-directed ligation. That seems to be more open. The two are rather different in their architecture, in their RNA structure, their domain structure, and their relation to peptides. There is an interesting thing that, for the sake of time, I skipped over in the slide. The structural center in the 2014 paper by Petrov et al. Is not the same as what we think of as the functional center in either the large or the small subunit. In the large subunit, it's the peptidal transfer center. I forget what it's called in the small. They choose their phases to start with a functional center. I'm curious whether we can revisit that and find out whether the structural center could be a good place to start. And we see even those earliest functions as hitchhikers in a kind of a hailstone world. All open. All of this needs to be turned into proper hypotheses and then tested through a combination of bioinformatics and actual synthesis. Thank you.
Thank you so much. Unfortunately,
Oh, that's the presentation. The whole video. Yeah.
Pretty awesome.
Pretty awesome, yeah. And at the last point, he mentioned about as he mentioned before, the layer of the ribosome is an onion, and the layer that is required for the two subunits to interact and perform translation, that layer became later. So this indicates that the two subunits, before they acquired this layer, were separate and doing their own separate things. And then, like, the last subunit was just a condensation catalyst, basically, and not just condensing amino acids into peptides, but also making polyesters, maybe even catalyzing polynucleotides, basically being a catalyst to make more RNA for itself. It's interesting too. Interesting possibility.
And they've put more meat on the bones of that hypothesis with their work on proto-ribosomes. There was a paper where they used basically like stripped down ribosomes, just working with the, if I'm remembering this correctly, the peptidyl transferase center in the LSU, and showing that out of the 13 they synthesized, 5 of them were capable of bond formation. So
right.
it looks, I think that was pretty good corroboration for what they're suggesting there.
It's funny that you mentioned that, because it's actually one of the notes I had prepared for this. I can show my own personal presentation. It's not too long, I hope. Like we're now at 1 minute 22, but I think I can get through this within 20 minutes or something like that. But if it's fine by you,
yes, please.
I can show my All right, sharing.
Yes,
my.
Please do.
All right, sharing my right now. There we go. So I had a few notes prepared about the origin of translation. One is the chemical context of the polymer world, the Darwinian threshold, RNA world ? And the eigen error threshold, and also a few things about the proto-ribosome and early polypeptides. So regarding the polymer world, there is especially the RNA first proponents. They will often make the claim that they often make in my opinion, the mistake that polymers such as polypeptides and polynucleotides, they were the inventors of catalysis, and because they were the inventors of catalysis, they were required as the first things of biochemistry. But this is my mistake. This is, in my opinion, a mistake, because polymers inherit catalysis from cofactors and coenzymes, not from themselves. For example, this has been noted by the metabolism-first proponents. They will, they note that in key reaction in core metabolism, it involves proteins that mediate these reactions in core metabolism. They involve metal clusters, which are reminiscent of minerals. And so the claim of metabolism-first proponents is that mineral catalyzed geochemistry gave rise to protometabolism, and this chemical context remains preserved to some extent in proteins by unfolding these geochemical mechanisms. Just interrupt me if you want to say anything, that's all.
Totally on board so far.
All right. But of course, metal clusters are not the only cofactors present in proteins. You have also the organic cofactors, also called coenzymes. And an interesting thing about them is they are somewhat like biochemical intermediates between the small metabolites of core metabolism and the larger polymers. For example, some of these coenzymes are direct building blocks of polymers. For example, ATP and GTP is, of course, the direct building blocks of RNA. They also have common nucleotide moieties between them. And they also exhibit the beginnings of polymerization. But they are not as pure or clean as the polymers themselves. For example, they are heterogeneous. So some cofactors are like polymers between both RNA nucleotides and amino acids. For example, the coenzyme A is built from cysteine, ATP, and beta-alanine, a form of alanine that is normally not found in the genetic code. Like in the genetic code, it's alpha-alanine, and a different isomer of that thing. And in these cofactors, you have also similar chemistry, like the same ester bonds present that are present in RNA chains and the amino acyl transfer. It misses T, but it should be tRNA. Yeah. And I think Morowitz, or maybe somebody else, I think Morowitz has called them chimeric polymers. Basically the beginnings of polymerization, but not in a very clean way that became later. So it's another argument for why cofactors and coenzymes preceded the polymers that we are known from RNA and proteins. And one last thing about the polymers is that even although more than half proteins, like over half proteins, require coenzymes or cofactors, but even the ones that don't require cofactors, they still often use these special amino acids like histidine, which seems to be amino acid or cofactors in amino acid form. Like histidine is produced by this pathway that produces cofactors important in catalysis and redox chemistry, and also, of course, the ATP and GTP. But histidine seems to be very chemically similar to these things, although it is turned into a amino acid, which is basically how proteins inherit catalysis in another way besides just unfolding these metal centers or coenzymes. Okay. Oh, and another thing about another thing about the genetic, even the genetic code itself seems to echo metabolism itself. There is a strong correlation between the letters of the second letter of the nucleotide is correlated with the properties, like some hydrophobic is correlated with one letter, and hydrophilic is correlated with another letter of the second codon. But the first codon, or the first letter of the codon, is correlated with the biochemical precursors which are found in the carboxylic acid cycle, basically. Like you have pyruvate, which produces these amino acids that are encoded by codons beginning with uracil. All right. Anything else to say so far, Trace?
Nothing just now, no.
All right, so moving on to the Darwinian threshold, which is like in the just pure evolution for now. Like there used to be a controversy surrounding the discovery of horizontal gene transfer, and you have basically one guy called Fourth Geosphere. He championed the concept or the counter concept against the concept of the tree of life where you have like a bifurcating tree relationship between living organisms. What Doolittle was basically saying is like, oh, since you have horizontal gene transfer, there can be no such thing as a tree of life. It looks more like a web of life, basically. However, there are apparent barriers against horizontal gene transfer, and this relates to the argument made by Carl Woese, where he championed the concept called the Darwinian threshold. Before what he's basically saying is that before the Darwinian threshold, horizontal holds free reign. And because horizontal gene transfer holds free reign, there was no preservation of vertical descent. So you have no pattern, no tree-like phylogenetic pattern, basically. And Carl Woese, I think it's called the progenode, basically. The progenode is when it's a phase of life where you have no barriers against horizontal gene transfer. But once you set up barriers against horizontal gene transfer, that's when you pass through the Darwinian threshold and you get these tree-like patterns. And the strongest signal of vertical descent that's still preserved is found in the nearly universal set of proteins or genes, the nearly universal gene sets, and most of these nearly universal genes that are found across all life are involved, unsurprisingly, in translation, the ribosomal components, and also ribosomal proteins. And these genes are most resistant to horizontal gene transfer, which preserves this, here I go. Trees built from these nearly universal genes don't agree on everything, on every relationship. So they are not totally immune from horizontal gene transfer, but they tend to agree on similar relationships more often than expected from random chance or from horizontal gene transfer bias, which you can get something similar to this from these special genes. So there's still a statistical tendency to preserve this evidence from universal descent, despite the interference of horizontal gene transfer. So why is the translational system resistant to horizontal gene transfer? Well, it has to do with how tightly this system is interlocked. Basically, we all know this system from our high school. RNA makes peptides, and peptides makes RNA. RNA performs the lytic function and chaperone function, and peptides they feed back on RNA by stabilizing, like in the ribosome. It's made of RNA, but the ribosome is also made of protein, of course, and the protein holds more of a stabilizing function. Like it stabilizes the RNA of the ribosome to perform more accurately. I think some researchers have made a naked RNA without proteins. It's still able to perform some catalytic function, but it's certainly not as accurate in translation and as stable as the normal ribosome. So this system arguably underpins the Darwinian dynamic of survival and reproduction and such. So. Anything so far, Drayce, to note?
I really like your presentation so far. You put together some good PowerPoints.
Oh, thank you. Yeah, and one last note is like the reason why this system is resistant to horizontal gene transfer is because this system is very, very tightly interlocked. Like you have many different components, and some, like especially in the ribosome, you have components that need to fit pretty well with each other. And thus, if you exchange a ribosomal gene between, like for example, a bacterium and archaean, that often fails because there are slightly different differences between the ribosomal genes, such that if you interchange them, it reduces the translational accuracy and such. So this is why horizontal gene transfer with these genes don't survive for very long, and this is why you have this resistance observed in these genes. So So as I say, there's a strict reciprocal relationship between polynucleotides and polypeptides. This chicken and egg relationship is often solved by proposing an early phase of life's origin with network level self-replicating RNA that was peptide independent. However, is this really the right solution? How would it explain the current translational system? Like if RNA was separately functioning from peptides, so at what point, why would you have this system where peptides feed back onto RNA and RNA feeds back onto peptides? Which is what you need to explain. Even if you are proposing an RNA world, you still need to explain why this relationship came to be. And maybe this relationship was established early on, which is what I'm arguing for why this relationship was established early on. Hold on, I need to drink something. My mouth is getting really dry right now.
I purposefully made sure I had some water, and I'm all out. I should actually replace mine soon.
All right.
Please continue.
Sure. There is something called the relevant to this conundrum is the something called the eigen error threshold. Basically, there is a limit to how much a genome can withstand the number of mutations per cycle. Basically, the larger your genome, the lower your rate, or the lower your mutation or your error rate needs to be. Otherwise, you get into a situation where you go extinct, basically, at a certain mutation rate. Like and the eigen paradox, again, quote unquote paradox, it says that without error correction, the max size of a genome is around 100 base pairs. However, error correcting complexes, like error correcting proteins, like polymerases that hold error correction properties, they require genomes larger than, or sequences larger than 100 base pairs to be encoded. Of course, this paradox is already broken by Eric Smith's proposal of the difference between folding and encoding sequences. So yeah, but there's more to be said about this paradox. Another solution to the eigen paradox is the Darwin eigen cycle proposed by Koonin. I think you also are familiar with Koonin as well, Thijs.
Yes.
And do you also know anything about the Darwin eigen cycle as well, or not yet?
Very little on this.
All right. So to quickly explain this idea, the Darwin eigen cycle, he proposed that without replication fidelity, you have a minimum number of genome size, but if you somehow improve just by the genetic drift, by like if you get a new function just by chance, you can increase fidelity through selection. But by genetic drift alone, you increase the size without a detrimental, without being detrimental, because you have increased the fidelity before. So you're allowed to increase in size through drift, which allows you to acquire a new function, increasing fidelity, and the cycle continues on. So basically, life can basically increase genome size and replication fidelity by climbing along this what Koonin calls the eigen cliff, or without falling off this eigen cliff. If you fall off the eigen cliff, then your genome size is too large for your replication fidelity. But as long as you stay above this line, then it's all fine, basically, what he's saying. All right. Oh, I need more water, so I will be right back real quick.
No problem. I think that's a pretty good use of neutral theory right there. As Nestle gets some water, I'm just going to talk about a recent paper, I think 2021, has basically looked for RNA selection in metal-doped clays. maybe later I'll pull up the paper, but regardless, if you put in ribose and metal doped clays in a Google Scholar, I'm sure it'll pop up. I think it's a 2021 paper. And what it shows is that ribose is selectively retained as opposed to other sugars in metal doped clays, and it has the kind of reactivity that you want, because there are other ways to stabilize and select for ribose using things like borate or silicate, but when it's adsorbed onto things like borate or silicate, it's less reactive. You want it to stay reactive to use it in further processes, and within and it's shown in this paper. Within metal doped clays, not only is it selectively retained, but it's also reactive to the point where in the paper they demonstrate the formation of nucleosides and then nucleotides, so they go all the way. And they also show that they return to it 42 days later, and the ribose is still stable, which anybody who knows anything about ribose degradation and how quickly it decays, how short its half-life usually is in bulk aqueous medium, will know that 42 days is a pretty good improvement upon a lot of other scenarios where ribose is a lot more unstable. And so I thought that was a pretty good paper. There are many papers coming out at rapid speed these days that are making a lot of progress. It's always interesting to hear people describing this field as not making any progress. The truth of it is that today abiogenesis is experiencing a renaissance. It's been doing it for about the past 15 years, and it's going strong. So a lot of really interesting stuff going on in this field.
Right, exactly. And then thank you for filling in the time when I was away getting water. Thank you.
No problem.
Picking off where I left off, so you have the Darwin eigen cycle. One thing is the question is how could the cycle have started? Like you still need some form of fidelity basically to increase to kickstart this process. Basically, it's this feedback loop of neutral evolution where you increase fidelity and genome size back and forth. But related to this eigen threshold is the error rate of protein synthesis. Like we are often familiar with mutations. Like if your DNA gets replicated, and there's sometimes an error, you inherit mutations. But there's also an error rate when it comes to which is less. There is less attention paid on this error rate in protein synthesis, where you can have errors in transcription and translation. For example, RNA polymerase has this error rate. tRNA, like sometimes transfer RNA can be mischarged with the wrong amino acid, basically. But by far the biggest error rate is the ribosome itself, where you can have frame shifts during translation, basically, where sometimes a codon, sometimes the codon gets skipped around or something like that. So something happens with the during translation, basically, and the error rate of protein synthesis is surprisingly high. Like it's between 1 in 10,000 or 1 in I think it should be 1 in 1,000. I think not 1 in 100. I think so. Yeah, it's like about like 1 in 10,000, 1 or 1 in 1,000 of every amino acid is wrong because of the error rate. And it's pretty astounding, because what this means is that 15% of all proteins in an E. coli bacterium has at least one missense error, like one misincorporated amino acid. And to name one protein in us, like our muscles have a particular protein called titin, which is a pretty long protein with over 34,000 amino acids. And given the error rate, on average, this protein has 17 errors on average. Like every single protein of this that we produce has this many errors. So given this error rate, why doesn't life spiral down in a self-destructive feedback loop wherein mistranslation produces broken error correcting proteins, and because of the broken error correcting proteins, it increases the rate of mistranslation, which increases the rate of broken proteins and such. Why doesn't this feedback loop get started? Well, basically, is these errors get buffered by two things. First, proteins have something called robustness. They are capable to fold and function properly, even though they experience a few errors occasionally, like as this figure shows. Like you have a brittle protein and a robust protein. Both of these experience the same number of errors, but the brittle protein often gets broken. It doesn't become. It gets non-functional very quickly. But the proteins, they stay functional despite these errors, basically. And another thing is the genetics code itself is structured in a way that buffers these errors. Like if you have a mutation, a mistake between the codon and anti-codon pairing, very often a similar amino acid gets still in a mistranslation. Sorry. Even if you have the wrong amino acid, often this wrong amino acid is still similar to the right amino acid, such that it preserves the overall function because it has similar properties. And that's why the genetic code is structured in this way, where similar, like for example, as I mentioned before, where you have hydrophobic amino acids sharing the same second letter of the codon, and the second letter of the codon is not coincidentally the most important codon because it's the most reliable codon where the least amount of errors occur. That's just the second codon, and the most unreliable codon is the third codon, which is why the genetic code relegates redundancy onto the third base pair of the codon, and it's a wobble base pair. Yeah. So my opinion is that because of this error buffering with robustness of the proteins and of polypeptides and also genetic code, my opinion is that error buffering can only happen with this RNA peptide relationship, which means that this relationship had to be established early on to get this error buffering going, to get this error buffering going. Yeah. I don't know if you have anything else to say at this point, Thijs. Please feel free to make comments.
Kunin has a paper on this about upon the third codon and wobble managing redundancy and its implications for the genetic code being capable of like a more robust response to error, and I can't remember when it's published, but people can probably look that up if they put in like Kunin, genetic code, and redundancy. I'm sure it'll pop up somewhere.
Yeah. Although the third base pair is reliable when you talk about pyrimidines or purines, right? So that is why, for example, arginine shares the purines, the A and G, and so serine shares the only the pyrimidines, the uracil and glycine. No, what's G C again of the nuclear base?
Which one?
Oh, what's C again? I forgot the nuclear.
Cytosine.
Oh, cytosine. Yeah, I mistakenly said glycine. It's cytosine. Yeah. All right. But yeah, it's the third codon is still able to distinguish pyrimidines and purines, but it's not as accurate as distinguishing adenine from guanine, for example. Which is why you can still have these 2-on-2 relationship with the third base pair. All right, so yeah, my opinion is that error buffering, this error buffering can only if for example, if you have an RNA-only system, perhaps there is something you can have error buffering, but I don't know. I don't know. Maybe there is a possibility, but I would say that if you have a cooperation between RNA and peptides, then you can have this error buffering going on, which prevents this collapse, basically, where you have this positive feedback. With the presence of these error buffering mechanisms, then this collapse is prevented, in my opinion. And so this comes back to what Eric Smith also talks about in his book with Morowitz. Is it prior to reliable translation, the only peptides and RNA sequences that can be made are statistical. Basically, you cannot ensure the same sequence or a very similar you cannot ensure the same sequence is replicated over and over again or produced over and over again. So what you have instead is a distribution of similar sequences which have an associative function overall, a statistical sequence, basically. As long as they don't have to have the exact same sequence, as long as they have roughly the right sequence. Sometimes there is a very large degree of leeway. As long as they have the sequence, then they can have a function, or maybe they can fold, for example, as to call back on the presentation with Eric Smith, perhaps can still fold nonetheless in this statistical nature. And statistical proteins, they have the unique characteristics that they are able to tolerate different translational contexts. For example, if you right now, if you change our genetic code, like a codon now codes for different amino acids, it is very damaging because now that error propagates to every single protein in our body. But with the statistical protein, RNA, if you change the genetic code, for example, now it's no longer as damaging as it normally is. And this statistical nature permits the free exchange of translational components and altering the genetic code to incrementally improve error buffering over time. And since this free exchange or horizontal gene transfer, this free exchange between components to improve the overall process is beneficial, everything converges on the same genetic code. Because if everything converges on the same genetic code, then horizontal gene transfer is able to occur more freely and more able to improve the system itself. So there's again a positive feedback loop between increasing the rate of horizontal gene transfer, which improves your genetic code or improves your system, which again allows for more horizontal gene transfer to occur. But then after you increase the reliability of replication or translation, whatever, this allows for selecting strictly encoded proteins. Although even the encoded proteins retain some statistical aspects, like the robustness against some degree of error of translation that I mentioned before, and at this point, genomes are able to get more information without falling off the eigenerror threshold. However, once such proteins appear, so these strict, like non or less statistical strict proteins appeared, then they become integrated. Further exchange of translational components becomes detrimental. Now the genetic code and the whole system freezes in place, and horizontal gene transfer is inhibited. Now you pass the Darwinian threshold, and this process is more elaborated in this citation right here, where horizontal gene, like free exchange between components in horizontal gene transfer in this early stage before the Darwinian threshold is important in improving translation and transcription and replication. So what a brilliant analogy that Eric Smith brings up in his book is the translational system, and specifically the ribosome, seems to be like a Wittgenstein's ladder, where you have processes that were necessary before, but when these processes allows you to improve something, then at that point it becomes necessary to get rid of the previous processes or previous things. So you climb up the ladder, but then it requires that you remove the early steps of the ladder to not fall back basically on the whole system. So it's a very good analogy.
That's awesome. I didn't think I'd ever hear Wittgenstein quoted in a biogenesis lecture. That's pretty awesome to see the Tractatus being referenced here.
Yeah. So Wittgenstein says that you need to learn wrong thing, or not. You need to understand me, and eventually realize that I'm nonsensical when he has used them as steps to climb beyond them. Yeah, it's a very good analogy. Like the whole system of peptides and RNA, they were more statistical, and the statistical nature was required to improve the genetic system. But once the genetic system of translation and such were improved, then you need to get rid of the statistical nature to inhibit the same processes that allowed you to improve the whole thing to begin with. So it has a reminiscent nature of the evolutionary ratchet, basically. Like you're only allowed to move in one direction, but once you move in one direction, you no longer can go back in the same direction. You are stuck in that place.
I want to give you a warning, Neslik. This might have a 2-hour limit on us, and we're approaching it. So I wanted to give you a heads up. Make sure. That
I hope I can finish.
Go ahead.
Expert has mentioned about this before, about the ribosome, where it shows your younger and older phases. And the polypeptides interact with these phases are cyclically different. The oldest are like random coils, and they are scaffolded by RNA in terms like these oldest proteins seems to be more the statistical proteins that I mentioned before. And then once translation was improved, you're allowed to have this complex and sophisticated folding. So I will go to this presentation real quickly to make sure we finish in time. Consistent with random peptide libraries, the oldest peptides tend to be associated with RNA, which stabilizes RNA. Like it's also funny that RNA is often treated as unstable in water, but when you look at, for example, the ribosome or RNA complexes that are stabilized by proteins are more able to survive for a lot longer time. But I think ribosomes last for a very long time in our cells as well, without being degraded. And proteins acquired autonomous folding later by achieving basically early proteins are like miniature folds that are not able to fold by themselves without RNA, but late proteins by duplicating or decorating these parts, they were able to fold autonomously without RNA as a scaffold. And again, this is what you mentioned before. This is the core of the large subunit, which seems to be a protoribosome formed by a homodimer of a 100 base pair structure motif, an ancient RNA fold, also seen in tRNA and ribozymes. And they scientists here, they reconstructed this homodimer with two RNA structures, which is able to mediate peptide bond formation. This is what you mentioned before I started my presentation. One last thing about the AARS. They show a peculiar relationship where class 1 AARS and class 2 AARS bind to the opposite sides of the transfer RNA. It seems to indicate that they used to bind to the tRNA at the same time, and then later on, when the genetic code expanded, they acted independently, which is a very interesting mechanisms for how the genetic code was expanded before. And in my opinion, this is the most well-supported and detailed phase of the origins of life, mainly because the ribosome contains a record of its own origins. And I'm done, just in time.
That was an excellent presentation, Neslik. Thank you very much.
All right. Any more quick remarks before we reach the time limit? I hope we don't reach the time limit, but here there is.
Yeah, I guess the last thing I'm going to mention, it's some recent stuff that's come out. There is discussions of like production of ribose with the foremost reaction and how it's not really ribose specific, but there are other sugars that are produced much more easily, like ribulose and fructose. And what they found is that under very mild conditions, these sugars like ribulose isomerize to ribose, and so it's actually a steady supply of ribose from these sugars that are synthesized in much more significant amounts. So that's like another route to ribose formation besides like the foremost reaction. And last thing is, and it looks like we're still recording, so that's good.
Yeah, I think so.
Is Laurie Barge just came out with a paper this past month on surficial hydrothermal vents. Especially, I found the paper about one of the two sites that she discusses just a week before I read her paper. It's a hydrothermal site at New Caledonia, and when you read the official paper describing it, it just looks like such a perfect recipe for everything that you need for like an emergence of life site. So just to push Laurie Barge's recent paper, and also if you look up the hydrothermal site at New Caledonia, sites like that I think are really promising, and it's a really good pointer to those people who continue to just talk about hydrothermal vents as this very static, narrow kind of environment located on the bottom of the sea with only like a very distinct set of environmental parameters, when in fact these systems are extremely variable, and so they can host all sorts of different kinds of chemistries. And it's really important to keep in mind, because there are straw men flying all over the place about that approach to abiogenesis. So
I often imagine, I think this is one of my questions that I posted to Michael Russell, is that when at the time, like 4 billion years ago, or more or less, at the time the moon was a lot closer to Earth, and I would imagine that the tidal forces on the rock itself would have influenced these hydrothermal systems, perhaps making the hydrothermal systems act like a PCR reactor, where sometimes temperature goes up because the moon passes over, and sometimes temperature goes down after the moon has passed over. Maybe a random idea that I have. I wonder.
Yeah, these kinds of environments are just chock full of all sorts of different gradients, and it's at this kind of interface that the emergence of life becomes possible. So where you have extremely variable conditions all meeting at this interface, and you end up producing a diverse chemistry that gives rise to a great deal of complexity. And so, so people should really stop describing hydrothermal vents in such a narrow way and appreciate the variability involved.
Also, the hydrothermal system does not just begins and ends with the mount that you are able to see. It goes much further down, right, where you can have processes taking place in the system. It's a very large system. Perhaps it was like maybe not entirely, but maybe it's like a huge interconnected hydrothermal system that took up a lot of area on the early Earth.
Yes, and like that's a lot of the time when you see them discussing these kinds of gradients, like when they reference a thermal gradient, they'll usually just refer to the hydrothermal fluid in the chimney structure as it's flowing through versus the temperature of the outside ocean water. But if you trace that hydrothermal fluid further down into the serpentinizing cracks, it's much hotter down there. And so you actually have a much larger thermal gradient than people usually appreciate, and things like that. And I guess the last thing to say here is that in the serpentinizing cracks that occur underneath this vent, I see absolutely no reason why the cracks that are directly underneath this vent, anything that's getting synthesized there ends up through flow getting concentrated within the chimney. And it's just this, like, I keep seeing these researchers giving this ridiculous dilution problem to vents, as if like the chimney structure isn't a concentrating a structure, but that's like the whole point. And they keep missing that too.
I would imagine that there are, like, if you go deep down into the crust where serpentinization happens, I think, like, when water goes up into the rock, I think they will. It's almost like a lightning pattern where they converge onto one channel, basically, or maybe like several channels where concentrations can get really high.
Yeah, and,
like
since
a
we're
funnel,
dealing with
in inverse funnel, yeah.
Yes, and since we're dealing with cracks, we're talking about a system that even down there is pretty confined, and so we should expect, especially if things like green rust accumulate within serpentinized cracks. If nitrate makes it down there, you get ammonium that's going to react with everything that's being produced with serpentinization. So prebiotic processes should be going on there, and then delivered up to the chimney structure. At not all of it, like those cracks that are further around the periphery of the vent might just end up diluted into the ocean. But those flow channels through the cracks underneath leading up to the chimney structure, whatever is produced will get concentrated there.
Right. I think also, like you already mentioned, nitrogen, how it's able to enter the system as well. I would also imagine that maybe hydrogen, or nitrogen, is able to enter the system from the Earth below, right? Like, do you think that most of the nitrogen originates from the atmosphere at that point? What do you think, where the nitrogen comes from to feed into this system?
There's a recent PCE3 YouTube video on where basically, like, the nitrogen on early Earth came from, and they are still treating it as atmospheric and ultimately derived from particular meteorites in outer space a lot of the time. But I'm open to the possibility of it coming from other sources. I'm just unaware of what they would be.
Yeah, I don't know. Like, I would imagine, like, I don't know. I imagine that the atmosphere of the early Earth was produced by outgassing from volcanic systems and such, and maybe around the time when the origin of life, maybe there was still a significant amount of outgassing from the mantle in terms of nitrogen, and maybe that's one way of how hydrothermal systems got fed nitrogen in the first place. Maybe it's my speculation at this point.
Sure, sounds possible to me. I'd have to look into it more, though.
Yeah. Like, of course, another possibility is, like, you mentioned that perhaps nitrogen in the atmosphere gets reduced to nitrous oxides, and that lands or rains down into the ocean and eventually comes into the hydrothermal vents, right? That's the other way.
Yeah, that's Russell's approach. That nitrate and nitrite get accumulated into the ocean through, like, lightning discharge and stuff like that over tens of millions of years. And yeah, that works. The only thing, though, is that it was pointed out to me recently by Lori Barge that nitrite is too unstable to make it all the way down to the bottom of the oceans. And so you would mostly be dealing with nitrate. However, again, we're talking about variable conditions for hydrothermal vents. So vents are also occurring in lakes, in lagoons, in basins. And in these areas, nitrate would much more likely be a component of the system.
But then you, of course, start to butt head with Michael Russell, who thinks that the early Earth was an ocean world, right? That's to propose lakes and lagoons, yeah.
Yep, he would be against those kinds of things. I'm still open to the idea there being land in the Hadean. principally through Koranaga, and I noticed that when Dr. Russell gave his presentation on my channel, he was really focusing on the volcanic islands like the Hawaii's and Ontong Javas and all that stuff, but he didn't touch on continental crust and plate tectonics, and there's still a huge debate about just how early that onset. And there are a lot of geologists now saying that, yeah, plate tectonics can go all the way back to the Hadean, even like, going all the way back to like the mid Hadean. And so what you need for that is basically your ocean, and that's going to start. Yeah.
It is also like
Yeah, so,
a problem. It's also a problem of like how thick the crust was. Like if the crust was a lot thinner back then perhaps everything, like even continental crust, would collapse and get below sea level. Maybe like this is another thing to bear in mind.
Yeah, it's still something that I'm working on to fully understand. But basically, the way that Koranaga approaches it, there is this big issue about the mantle being too hot, and I want to check out more about that to see about cooling rates for the mantle over time during the Hadean, because initially Russell was arguing about magma ocean solidification taking like 500 million years and referencing that article by Montu. But then Koranaga pointed out that through the Rayleigh-Taylor ability, the cooling rate is greatly increased. And so maybe, maybe with respect to the mantle, it could have been cooler in the past. It's something that I would have to look into. But one last thing that's important about that is Russell also says that the ocean was never less than 4 kilometers deep. However, Koranaga's work on magma ocean solidification was suggesting that it's actually much shallower, given the actual way that magma ocean solidification works, where his model actually has it at 1 kilometer, and so then it becomes this race between the gradual rising of the ocean versus the rate of production of continental crust.
All right. And one last, like I think it's already been 2 hours. Maybe, or maybe if you want to talk more, I'm fine with longer talking longer. But one last thing that I think about is the phosphate. Like, is there also something about phosphate? How phosphate could enter into the hydrothermal systems?
So Matt Pasick and others who I can't remember off the top of my head, talking about serpentinization as a way to liberate reactive phosphate, and then also a very recent article on the availability of phosphate in the Hadean Ocean because of Fe2+. There's something with respect to the greater availability of Fe2+ in the Hadean Ocean that actually leads to like a level of available phosphate that's like 4 orders of magnitude higher than previous estimates, and that's something that was published, I think, just recently in 2021 or 2022. So for
Fe3+ is the rust iron, right? And Fe2+ is the
that's
green iron.
Yep. So you're talking ferrous versus ferric iron, and with your green rust, that has both. It has a distribution of Fe2+ and Fe3+ throughout its layers.
All right. And because of the Fe2+ iron, you have a more availability of phosphate potentially.
That's what this article was arguing. Yeah. Let me see. Maybe I can actually find it really quick. I'll look it up to just give you the exact title. I read it a while ago, so I'm forgetting some of the details, but it's a recent one. Let me see if I can find it real quick.
All right.
Filibuster for me while I'm looking.
Oh, all right. Maybe I can. I don't know. Like I went over my slides in the last few minutes really quickly. But one thing it's really interesting about the amino acyl tRNA synthetases. But the ones that I mentioned last about how the genetic code got expanded, these proteins, the amino acyl tRNA synthetases, they impose the genetic code onto by charging the right tRNAs with the right amino acids, and that's how the code gets imposed into the whole transcriptional system. Without these amino acyl tRNA synthetases, then the code doesn't work. And what they showed is that through duplication and perhaps even like separation between the two classes, and then that's how the code got expanded. Perhaps that's a very interesting thing to consider.
The paper is entitled "Marine Phosphate Availability and the Chemical Origins of Life on Earth," published by Brady et al., Nature Communications, 2022.
All right.
All right, Neslik, do you have any final thoughts for us?
I'm thinking. Let's see. I think I have no longer any thoughts, and I think it's also a very good limit to stop at 2 hours and 15 minutes. In case anybody else wants to rewatch this, they are not shocked by 4 hours. No way. So yeah.
Fair enough, Neslik, and thank you again for both reviewing this Eric Smith video with me and also for a pretty awesome PowerPoint presentation. You should put more of those together. And you were saying, I don't know if it was during our recording or beforehand, when you mentioned that you hoped you could deliver a presentation that was worthy or basking in the light of Eric Smith's presentation style. And personally, I'll just tell you that I think you were a stunning success. That was a pretty good presentation. I hope to see more of that from you in the future. Yeah.
Thank you. All right. And on that note,
all right,
it's a good time to end it. Yeah.
Yeah, let's do it. All right. So with that, we're going to end our recording, and this should either post to YouTube automatically or give me some direction on how to upload it later. I'll figure that out, but this will be up on YouTube, I'm sure. And with that, we're going to end our recording. I hope everybody enjoyed, and have a good night.